Okay, let's talk about HER2 and why HER2 worked as a target very well. I was way back, people from City of Hope in UCLA, they looked into HER2 and they found this HER2 is overexpressed and in the breast cancer. For example, this is the normal where you have typically 2 copies, and when you look into breast cancer tissue, they have found several copies of HER2, and I'm talking about genes, by the way. HER2 copy number on the chromosomes is more, and when your copy number in the chromosome more, it reflected the protein as well on the cell surface. I just told you, typically HER2 expression in the normal tissues somewhere like 50 to 100,000 copies, whereas in the cancer tissue is somewhere like 1 million to 2 million copies. So that's what is essentially, it's more copy number, more mRNA transcription, more cell surface protein, and obviously more of extracellular domain, and that's the logic, okay, why you want to work with the target. And this was confirmed in the tissues by in situ hybridization. The way it works is you label the RNA probe with the fluorescent dye and label your DNA, and it gives you how many copies you have it actually. So you can see the normal, in this case this is normal, you see the pink is lot more here, and the copy number is lot more in the cancer tissue, so does the protein. This is immunohistochemistry, IHC, and you do with the antibody here. You take paraffin expression, you see nothing, it's just nuclear staining you have with the nuclear protein staining here, but that's not HER2. And you see a moderate expression, this is high expression, and that reflected in the copy number 2. And it is actually both, I forgot, HER2 also overexpressed in breast, ovarian, and gastric. So actually it is 3 indications. Now that reflected in the cells too. What they did, MCF7. MCF7 is expression of HER2 is normal, and when you take HER2 gene, put into MCF7, you basically overexpressing it. When you overexpress HER2 in MCF7, what happens? The DNA synthesis is increased, cell growth rate increased, and colony formation of soft agar is increased, tumorigenicity in the nude mice is increased. So that's a typical property of an oncogene. You take an oncogene and overexpress in a given cell line, it becomes cancerous essentially. That's what is oncogene mean, and HER2 is an oncogene. And just a little bit quick on breast cancer, in the statistics in the United States, breast cancer is 230,000 new cases diagnosed every year, and unfortunately close to 40,000 people die every year of breast cancer. And but however, it changed, and it changed dramatically. We talked about ER positive, HER2 negative breast cancer, and tamoxifen, and there are many estrogen positive small molecule drugs actually did a wonderful job. So when you have ER positive HER2 negative breast cancer, it's not a big deal. People can still live without any treatment for several years. But it's not true with the case of HER2 positive breast cancer. The breast cancer you think about, there are at least, let me see if I have a slide. So the breast cancer you can actually classify into broadly 3. One is HER2 positive, other one is ER positive, HER2 negative, third one is triple negative. The ER positive and HER2 negative you can further, subdivide into luminal A, luminal B, and again further, but there are 3 broad categories. One is HER2 positive, that means HER2 overexpressed it. HER2 negative ER positive, that means ER estrogen or progesterone receptor overexpressed in that. The triple negative, both are negative, HER2 negative as well as ER/PR negative. That means neither of these. If you think about HER2 positive breast cancer about 15-20 years ago, whoever diagnosed with HER2 positive breast cancer, is basically they have been circled, you're out in next few months. It was that aggressive breast cancer it is. But today they are the most lucky ones because there is a targeted therapy, and actually people they lived without cancer for so many years because of Herceptin treatment. Of course they do surgery first, then do Herceptin with chemo. So it did cure actually a lot of people. So however, today is the biggest threat is the triple negative. There is no therapy as of today, and people still figuring it out. HER1 nothing but EGFR, and these are the downstream adapters and further signaling molecules, and it goes all the way down. And they modulate so many pathways such as apoptosis, migration, growth, adhesion, and differentiation. Where do we start it? We are working with HER2. Where does HER2 play in this role? And here in the case of HER2, HER3 for example, is heterodimer, and HER3 is actually a pseudokinase domain, means it has no kinase domain. Should have been labeled differently. So it should be HER2 and HER1. HER2 is a pseudokinase. It does not have an active kinase domain, but HER1 binding domain. Okay, that's one way of doing it. Or you can block the downstream, which is a PI3K inhibitor. You can use it. Or you could also use this kinase domain and use a kinase molecule. Use that to inhibit the kinase function. Or you can also use AKT, for example. So there is various ways you can approach this pathway. So for now we are talking about biologics. We already talked about it. It is ORX, again tell about how people are developed Herceptin in few slides later. And but that's not the only thing. But HER2 also pairs with HER3. It's ligand dependent. And there is another antibody developed by Genentech by pertuzumab, Perzeta, that actually you can disrupt this dimer formation, HER2, HER3 dimer formation, and use the antibody to disrupt that, and then by doing that you are actually inhibit the signaling pathway. So, and on top of that, they have Kadcyla, which works in the same way, but a different mechanism. Okay, what is HER2? We just talked about it. It's a receptor tyrosine kinase. It's a extracellular domain. You can see so beautiful, and then this is a transmembrane domain, then intracellular domain. Particular antibody only worked. That's very, very important. That's part of your screening. You make maybe 20-30 antibodies, for example. Not necessarily you will make 20-30 antibodies, depending on how big is your protein. If it's a too big protein, you can make a lot of antibodies. Small protein, you only can make 1 or 2. It all depends, because when you have a huge protein, you have a lot of epitopes. Then each epitope gives one antibody. But with a small protein, you don't have. So it all depends on the size of the molecule. So, but they have to screen a lot of antibodies to come up with a conclusion that so and so antibody works, and that so and so is Herceptin. HER2 overexpressed only in 25% of the breast cancer patients. If they didn't have the knowledge, what they would have done, they would have done clinical trial in all the breast cancer patients. The outcome would be what? 75% will not respond, only 25% will respond. Then what FDA will say is majority don't respond, I don't think this is going to work. So what they did is, based on the knowledge, they said they screened the patient up front, which patient expressed HER2. Because you have to know, if you have an antibody, if a protein is not there, what will antibody will do? It will not bind, right? So that way up front actually they did a clinical trial only on HER2 positive breast cancer patients, and because of that it worked. So the diagnostics comes into play, and I think if you think about this, the first approved monoclonal antibody of cancer with the diagnostic test actually. So let's walk through simple steps. So when you think about large molecule versus small molecule, again I said rational screening and random screening, rational design. Small molecule can happen both, large molecule can only happen rational design. Then we have 4 steps in the drug discovery development: discovery, development, manufacturing, commercial, and marketing. Okay, that 4 you can divide into 20 different aspects, but I'm just putting in a broad aspects. When you think about HER2 discovery, we ask these questions: what is the disease? In this case, breast cancer. Why does it happen? Because HER2 is oncogene. When your oncogene has more copy number, it supports the proliferation, therefore the cancer progression is super fast. So, and how does it happen? Of course, it has increased copy number, increased mRNA level, increased protein level, okay? And then what do you want to do with all that? I want to make antibodies and screen for the antibodies for HER2, which inhibits the HER2 protein activity. That is the key. For everything you want to do in your life in science, you got to have an assay. If you don't have an assay, what you want to do, you don't have functional assays to screen, you are no go, you cannot go anywhere. That is the first step in science. You got to first, how you want to screen, you got to have an assay to screen. So in this case, you can do proliferation, you can do in vivo tumor growth, there are multiple things you can do. That's what has been actually been done. Then development, that's a discovery. I am hoping that all of you got it step by step what we are doing here. We are done with the discovery. Now what you did, You know why it works, how it works, mechanism of action, and your goal is to inhibit the HER2 function in the cancer, right? And you, for that you made antibodies, you screened the antibodies, you found an antibody that inhibits the function of HER2, which can stop the cancer. You are there. You are not done yet. That is the first step. Okay? Then you do development. When you say development, that is two aspects, preclinical and clinical. So, when you think about preclinical, what is preclinical? Basically, you do animal studies. Hey, you showed in vitro in cell based model it works. You made antibody, you screened it, you show that it inhibits the cellular growth. Now, what do you do with that? You have to develop models, breast cancer models you have to develop. And obviously, for that you do, typically you take a cell line which has the potential to grow tumors in the nude mice. In that case, you take a breast cancer cell line, ideally which express the HER2. You can also take a cell line which does not express the HER2. You have to have a control for every experiment. Now, you take this antibody and then you first immunize, you take the cell line and you inject the tumors and let the tumors establish. Then you go and inject this tumor bearing mice with this antibody and a control antibody which does not bind HER2. Okay? You have to have a control. And then you see what happens. The tumor should shrink, tumor size should decrease. Okay? It does not happen, there is no goal. And you take the same antibody, put it in a non-HER2 expressing breast cancer model, it should not do anything. That is why you establish it in vivo. Once you do that, that is the same efficacy. What about safety? Safety you do in two different models for biologics, but I can quickly tell about it in a minute. You take the antibody, you do in rodent models first and see, you dose the rodent mice or rat and dose and see what happens. The normal mice I am talking about. This antibody should not do anything typically, because if it does something then it is bad, but at what dose it does. Okay, it is efficacious, it has a potency at 1 microgram. If the mice or rat can only tolerate to 10 mg, do you worry? That is 1 microgram versus 10 mg? No way, I mean you are not going to worry, even tenfold is huge. So, but if it is only active 1 mg, your safety also 1 mg, I do not think you want to proceed it, because your efficacy as well as your potency as well as safety at the same dose. So, you will not have the window to work with. But once you establish that rules and data in the small animal model, then you go to the next level, large animals, in this case monkeys, and you got to do that, and without that they will not let you do the human clinical trials. So, you take, you have an idea now. In the animal models, let us assume 1 mg per kg it works, it can actually you can have completely eradicate the tumors and the cancer gone. By the way, it is easy to cure cancer in mice, not in people. Okay? So, every cancer has been cured in mice so far. So, animals are animals, end of the day, it is the model, right? But you can say, then why are we doing it? If it does not work in mice, it is not going to work in human. Okay? So, that is the, so at least you show that it works there, and if it works then you take next level. Anyway, the safety models in monkeys you do it, you have an idea, let us assume it 1 mg per kg worked, it was safe for 20 mg per kg in mice. Then now I want to show in the animals whether it is safe for 1, 3, by the way it is mice in 20, I will not put 20, I have to slowly inject and see what is the tolerability in the animal models, in the monkeys. And then you determine, okay, up to 10 mg per kg in monkeys is safer. You got an idea. If you dose anything more than 10 mg per kg, it will be a problem. The one more thing is determining the tox species is important, is that protein must be expressed in that species that you are working with. In this case, HER2 should be expressed in monkeys, number 1. The antibody that you developed should cross react with the monkey HER2. You get it? The logic? It does not cross react, what happens? You cannot determine the toxicity because it does not bind. Okay? That is very, very important. The determining the tox species, your antibody must cross react with the species that you are working with and also should work in human. Well, I only have an antibody works in human, does not have antibody that works in monkeys. What do? You make an antibody works that in monkeys, that is called surrogate antibody, and show that potency of the surrogate antibody as good as the human equivalent, and make a model for that. You can do that actually in the rare situation, but it's double work. You just imagine now you have to do project 2 times, and money and all cost and effort and all that. So that's why you make the tox species. So that's the preclinical. Once after that done, you actually submit a package to FDA that I want to do clinical trial. That's called IND. Okay. So you submit a package with all this data, discovery, preclinical data, and show that this is the drug we developed. Now this is the drug, it works at this dose, and this is the safety dose. Now I want to work in humans to figure it out what is the safety dose in the human. That's called phase 1 clinical trial. The goal of phase 1 clinical trial is actually to determine the maximum tolerated dose in humans. Typically you do in 20 to 100 patients depending on the, this is for cancer, okay. Every disease indication is different. For cancer, that is the goal. You do, depending on your objective, you do 20, some people do with 100 depending on what you were looking for. Then you go to phase 1 is safer, you find out so and so. Let us say in monkeys 10 mg per kg is safer, most likely you end up with human is 5 mg per kg, twofold, because we are big. So, and 5 mg per kg you find out. So what is our potency? 1 mg. Still you have a window to work with, 1 and 5, right? So now you go to humans and dose it from starting from 0.2 and dose up to 5 and figure it out if it's safer or not. Once you know it's safer, okay, now we have found it safer. And what you do, you take it to that drug and put in the cancer patients. Actually you can work with cancer patient phase 1 directly. You don't have to work with healthy individuals, but you ensure that people are not dying because of your drug. Okay, that is the key. Okay, once you ensure that, now you go back and look at where the people got benefited. So how do? You have to do repeat dosing. Now you figure it out, 5 mg is good. What I will do, I will go and 5 mg is safer, is okay, then what I will do, I will go lower than 5 mg and keep dosing once in 3-4 weeks and till you see some benefit, either tumor shrinkage or a life improvement or something you have to have. There has to be a way to figure it out. Whether quality of life is changed or tumor shrinkage happened, you do a lot of techniques, MRI and all that, you can do it. Once you got that benefit, that is in phase 2, you do that, about 50 to 200 people, then you do randomized. Here you already know when you are doing phase 1, phase 2, who has cancer, who does not have cancer, which patient got actually what drug, but when you go to phase 3, it is a random trial. You work with all breast cancer patients, but what you do, you do not know who is getting placebo and who is getting drug. Unfortunately, you have 2 groups. The people who are dosing know what they are dosing, but it is blinded for everyone else. So that is called randomized trial, because you do not want to be biased in your trial, but that is a huge number. If cancer is about 500 to and above, but if you think about metabolic disorder, they are to several thousands, because you have to show the safety of the drug. So for cancer up to 500 is more than enough actually to establish, and in fact there is unmet need. So it's very, very important when you are thinking about developing a drug, what cancer you want to work with. If there is a drug already for a given cancer indication, the bar is very high, you have to beat that. But there is no drug, for example pancreatic cancer, there is no drug. You start making money only when you get after approval. Before that you don't make money, you just consume money, billions of dollars. Okay? So you have to think about how to survive, and who will have deep pockets, who can fund you, and we can take all the way to that. I can only do phase 1, I do not have money and lift hands. No, there are people out there with deep pocket, they can fund you, they can give you money, they take your program to phase 3. The big pharmas, that is what they do. That is called mergers and acquisitions, collaborations, and antibodies. I just talked about Y-shaped molecule. This is what I meant when I say Y-shaped molecule. So this is the FC portion. By the way, this is light chain, this is heavy chain. This is what we are talking about, constant domain in the Fab. And these were the one we are talking about, CDRs basically, the little CDRs here at the end, tip of the antibody. So that is what you replace it in the human framework, you make it humanization. Or nowadays you can make, if you have transgenic mice, you can make human antibodies directly, but only few companies own the technology. You have to pay a lot of money to do that. So you can't do by yourself. So this is a Herceptin trial, and you can see this is this is what I am talking about, phase 3 trial. The orange one is the patients who got Herceptin with the paclitaxel antibody along with chemotherapy. whereas the patients only got paclitaxel, and you see a huge improvement and in survival. Okay, so again, there are many approved large molecule therapeutics, and as I said, for this pathway only I am talking about, and there are plenty of them other ones. Herceptin by Genentech Roche, Hybridization for anti-GFR, Vectibix, that's anti-GFR from Amgen, and Prezeta again from Genentech, that's for HER2 HER3 dimerization inhibitor. The TDM one again from Genentech. There is another one from Genentech, HER1 HER3 antibody, dual acting. Same antibody binds two different proteins actually, that's called dual acting. These are all the approved drugs pretty much and listed, and that's not the only table. You have, these are all antibodies by the way, and you can get it from that source that I listed there. But again, this is the principles and rules are pretty much same. Then another one is a different molecule called antibody drug conjugate. It's still antibody, but it's different. So what is antibody drug conjugate? So antibody drug conjugate is a three component you have: antibody, a linker, and a cytotoxic drug. You all heard about chemotherapy, toxic effects. When patient dose with the chemotherapy, what happens? It's a small molecule drug, goes all over the body, kills normal cell, kills cancer cell. Does it differentiate between normal and cancer cell? It will not, because there is no mechanism to do that. However, when you attach a potent drug to the antibody, what antibody does? Antibody only binds the cancer tissue. It does not bind the normal tissue. So because of that, now payload is being safely taken to the cancer tissue and dumped there. So because of the window in the normal payload, you see the efficacy toxic dose is very, very narrow, but because you attach to antibody carries, now window is broadened because the normal tissues are being spared. That is, the antibody does that job, a targeted delivery basically. And again, when you think about antibody drug conjugate, this is antibody Y-shaped molecule. By the way, these are the CDRs I'm talking about. So the rest all is variable domain. This is a variable domain, this is a constant domain, these are CDRs. So this is antigen is important. When you think about antibody drug conjugate, antigen is very, very important because that antigen has to be expressed only in the cancer tissue, and it should be minimally expressed in the normal tissue. Again, the rules are same. And then antibody you have to develop, it has to behave, it has to bind well, it has to have good affinity. And one important for this, we didn't talk about till now, internalization. Internalization, okay, internalization basically you have cell, right, cell surface. When this molecule binds on the cell surface, it has to get inside the cell. That's process called internalization. There is a process called receptor mediated endocytosis. Endocytosis is the word, typically we use it. So when the molecule goes inside the cell, it goes to endosome first, then it goes to lysosome. When it goes to lysosome, all protein get chewed up because there is lysosome is a degradative organelle. So it chews up all the proteins and antibody. Okay? So that's what I'm talking about internalization. I'll tell you in a minute what does that mean. So it's very important target internalize because when antibody binds on cell surface, it takes inside, okay. Once it goes inside, goes to lysosome, antibody is degraded, the payload attached to antibody is not degraded because small molecule cannot be degraded, right. So that's what the molecule has to go inside, how it will go, antibody will take it, how will antibody will take, antigen has to actually add that property. Every antigen you cannot have internalization on the cell surface, there are few will not internalize. So you have to figure it out that upfront for that. So then you have to know what drug you want to attach, then you have to know how you want to attach linker. So this is you have to think just beyond antibody, you know, all this you have to think about it when you are thinking about all this. So the Catzella is one such drug what was approved few years ago by Genentech, and many of us worked on it, and it's a successful drug, and it actually works better than Herceptin. Now I'll tell you why it works better than Herceptin. So when you think about this target, what kind of things you have to think about it? Now target validation for antibody drug conjugate is different from target validation for antibody by itself. It has to have special rules. First thing you think about is this toxic window, expression in the cancer versus expression in the normal. If they are same, it's twofold expression in cancer versus 100 copies in cancer versus 100 copies in normal, no. 200, 100, no. At least you should have 5-fold difference between cancer and normal. If you have 100-fold, that's fantastic, but you will never have that. Okay? So that's what we call window. So you can figure it out based on the big databases and all available tools through bioinformatics approaches. Then first thing you do is freedom to operate. Hey, I have this gene expressed in cancer very well, you can make antibodies, but can you make antibodies and own it? That's the first thing. These days no one can own genes. There 20 years ago people used to patent on genes, no longer you can do that. Okay? Only thing you can do, you can make an antibody and file a patent and you own that antibody, that's fine. But there are such situations that some people may say you cannot make antibodies that protein any longer. They may say antibodies made against this protein, we own it. There are some claims like that. You have to think about upfront whether you can make antibody freely and operate it in commercial purposes. Skyrocketed compared to green one, which is here. And all green ones are pretty much here. It is there in breast, it is there in ovarian cancer, somewhere I should see gastric. Here, this little stomach, few dots, but there are few patients. That's what this is US population. This is more enriched in Japanese population actually. So these are the three indications. That's why you figure it out from. These are available data actually for every gene. Actually, is available in TCGA now. You can just log into TCGA, look at the gene, and get this data in a few minutes. You already have data, whether the cancer is overexpressed or not. Then what to do later is the key. So again, here I know the protein overexpressed it. I know it's on the surface, I know it's internalized, I made the antibody which binds, I showed it goes inside the cell, I conjugated with the linker payload polymerization. If you actually rewind your cell biology class, tubulin polymerization is very important for mitosis. Basically, if you inhibit the tubulin polymerization, it inhibits the cell division. If you inhibit the cell division, cells will die, correct, right? Cell cannot live forever, right? So the way it propagates by doing the division, and then later on the cells dying, and you have to replenish that died cells through continuous metastasis, continuous cell division. So what you are having, you test this on the cells. You see this is SKBR3 cells, that's the HER2 expressing breast cancer cell line, and you put this antibody drug conjugate on the cells. You see only when it works is the antibody drug conjugate treated. Linker payload alone, antibody alone has very little effect. Free drug will work because free drug will go inside the cell and work. But most striking thing is this in vivo study, tumor. This is Founder 5 model where how to overexpress results in the tumors basically in the breast. This is basically it's the tumor tissues actually penetrate in the tissue. It's then you grow the tumors in under the skin basically. That's how it works. It's not a breast tumor. It's a breast tumor, but it's not in the breast tissue basically. So what the bottom line is, you treat the tumors with vehicle means antibody alone or trastuzumab alone or some other buffer control, then the antibody drug conjugate, in this case TDM1, you see only TDM1 works. Everything else, even you take the payload, mix with the antibody, it does not do it. Only you attach physically the payload to the antibody, it works. Okay, but simply mix the payload with the antibody, it does not work because antibody cannot carry it. It can only carry its physically attached. And in this case, antibody, you say Herceptin, it works for HER2 binding. It also works, unfortunately I have to use another term called ADCC in this case, antibody dependent cytotoxic killing. That's a different way antibodies function. It binds the cells, target cells, and kill the cells through antibody cell killing. And then it inhibits the AKT phosphorylation because I told you HER2, HER1 signaling is involved in PI3 kinase and AKT. One way of looking at it is AKT phosphorylation. When you incubate the HER2 antibody and it decreases the AKT phosphorylation, whether you HER2 antibody or antibody drug conjugate, both do that. And another thing is the ECD. The HER2 also, the ECD gets shedded. So that's another way to control the tumor growth in the HER2 breast cancer. So both happen, antibody as well as antibody drug conjugate. So here the data to show antibody drug conjugate works in various different breast cancer models.